Today we'll discuss on methods of breeding in vegetable crops, followed for improvement. And if you just look at what all the things, the methods of breeding available in general in the plant breeding: mass selection, pure line selection, pedigree method of selection, bulk method, backcross method, single plant selection, clonal selection, pure line family method, single seed descent method, recurrent selection. In that you have got simple recurrent selection for GCA, recurrent selection for SCA, reciprocal recurrent selection. Then the modifications of backcross methods like the double backcross method or reciprocal backcross method. Then backcross pedigree method, hybrid of the backcross and then pedigree. Then you have a progeny test and line breeding. The extension of that is family breeding. Then you have got heterosis breeding, ploidy breeding, and mutation breeding. Nutshell and the many modifications you can find, as the individual crops some breeders are handling. But these are broadly the methods of breeding. If you just look at these methods of breeding, there are, the methods suited for cross-pollinated crops and methods suited for self-pollinated crops. Right, self-pollinated crops, what methods are suited and what methods are suited for cross-pollinated crops. You can just get, and in addition to that, you can also look at these plant breeding methods broadly into two groups. One is methods to handle the segregating material and population improvement methods, right? So this also you can just look at nutshell all these methods. Mass selection, if you just look at, it's mainly followed in cross-pollinated crops and rarely in self-pollinated. Some exceptions are there, but maybe their ignorance or some immediate use of that method that they have used. But by and large, the advantage of using this specific method for whether cross or self-pollinated crop is very much important. Otherwise, you are wasting the resources. You will not be a very efficient plant breeder, tell you. Yeah, mass selection, because why this is, the mass selection means you are not selecting the individual plants and separating it out. So in a population, you select some plants and those seeds you are collecting and massing them, mixing them to raise the next generation. That's most important. So here, if you do that in self-pollinated crop, what will happen? The individual plants are different in self-pollinated crops. They are homozygous. They will not, they are not at heterozygous status. So individual plants. So homozygous means immediately one plant you can separate it out, and if it is very good, you can push it for releasing as a variety. But in mass selection, it's not like that. It is the gene frequency is important, because all the plants are heterozygous in condition, and there is heterogeneity on the plant, right? But depending on the characters which you are choosing, you are going to mass them. So this method is well suited. Mass selection is well suited for cross-pollinated crops. And then you got a pure line selection. Yes, this is the method suited for self-pollinated crop, because what is that base population use is a mixture of several self-pollinated genotypes. So in that, if you isolate some good plants, it will go as a variety straight away, not like mass selection. You have to keep on doing it for several generations. Then the improvement you can see because of the gene frequency and genotypic frequency improvement for the targeted characters. So here it is already, the plants are, straight away, they're available. Only have to identify the good one and separate them out, because individually plants are homozygous in condition. So it is, say, pure line selection is well suited for self-pollinated crops. Pedigree method of selection. This is whatever, the pedigree method means there are methods which are used for handling the segregating material. That means you have to cross two parents and then you get F1, and from F2, self the F1, you'll get the F2. From F2 onwards, you keep on selfing, and that the plant, all the plants are segregating, right? They are separating out as per the Mendel's law of segregation. That the hybrid, if you self, it will start segregating. So handling that segregating material, you have got some methods like pedigree method of selection and the bulk method of selection. So bulking, pedigree means each and every generation you have to separate it out and keep the record of that individual plants. Whereas bulk method, no, it is just like mass selection only, but this method is suited for self-ponded crops because you are making the crosses and you're simply collecting the seeds, right? It is naturally self-ponded crops, they'll get into homozygosity, right? But whereas in cross-pollinated crop mass selection, random mating is there, it's only gene frequency is going to matter. Whereas in bulk method, you are crossing two parents and getting the F1 and then selfing the F1, and you keep on, the bulking, not separating individual plants. Bulk, right? So this is well suited for self-pollinated crops. And similarly, one more method of handling cross-pollinated, the segregating generations is a backcross method. So backcross method, it is of course you donor parent and recipient parent you are crossing, and the recipient parent you are using it as the repetitive parent or recurrent parent to gain all the characters of the recurrent parent. So it's also handling the segregating material. But most importantly, this is used for transferring one or few characters from one genotype to other genotype. Because it's not selfing in the subsequent generation, it is the backcrossing is involved. Whereas in pure line, in the pedigree method or bulk method, it is selfing only, right? So then you have got single plant selection. Of course, some good plant, if you encounter, it's not really a plant breeding method, you can list it here. I tell you, because a good plant, you select and separate it out. You can push it as a variety. Again, this is going to be successful in self-pollinated, not in cross-pollinated crops. Then clonal selection. Of course, this is the method for the asexually propagated crops. Some clones, you have to use the mixture of clones. Several clones you have to test, and which is the good clone you are going to release it as a variety, clonal selection. Then pure line family method is there. It is, again, you have got some certain modifications like your pure line method. It's a pure line family method. Then single seed descent method. It is again a modification of your pedigree bulk method. Rather, modification is there. It is handling the segregating material only. Single seed descent method, right? Pedigree, bulk. We'll discuss some of these methods in the next few classes. And then I got this single seed descent method. Again, it is handling segregating material. Definitely, it is again well suited for the self-pollinated crops. Then we have got recurrent selection. This is the method like mass selection, right? It is suited for cross-pollinated crops. Right here, mass selection, simply you're selecting the plants and mixing the seeds and for the next generation raising. But here there are some deviations, some crossing is involved in recurrent selection. That is a deviation, right? So again in recurrent selection, you have got four different schemes like simple recurrent selection for general combining ability, recurrent selection for specific combining ability, and reciprocal recurrent selection. So this is well suited for cross-pollinated crops. So it doesn't mean that it's only in cross-pollinated. You can do it in self-pollinated crops also with some specific objective. Like, if you want to go for recurrent selection for GCA and SCA, you want to improve in self-pollinated crops also. Of course, GCA, SCA is more meaningful for the cross-pollinated crops than the self-pollinated crops. In that case, you can say, but still there are, the hybrids in self-pollinated crops. Heterosis breeding is also used in self-pollinated crop. So this scheme can give the material for that heterosis breeding, right? Whether it is a self or cross-pollinated crops. So yes, generally, recurrent selection suited for cross-pollinated crops. So don't always think about the exceptions. Yes, you can do it. You can deviate from the normal rules, but you should have some advantage there. If you don't have any other ways, yeah, you can use them. Then you have got modifications of backcross method. Again, these are the handling segregating material only. Double backcross method or reciprocal backcross method. We'll discuss backcross pedigree method, some modification. Then you have got a progeny test and line breeding. Again, this is going to be for a cross-pollinated family breeding, some advanced way of the line breeding, family breeding group. the plants you can, the family you can have. That means the separate, several lines you are going to use than simple line breeding. This is again for the cross-pollinated crops. And of course, heterosis breeding is very important, which is feeding the whole world because of the heterotic hybrids. They are high yielding in character. So this method is very important, right? So heterosis breeding, hybrids are very popular and they are very high yielders compared to the open pollinated varieties. That's why heterosis breeding is very important. Even in self-pollinated crops, it is having the role. Then ploidy breeding, the crops which are, asexually propagated, or, you have got some specific ploidies changed by altering the ploidies, you can get the improvement. Yeah, ploidy breeding. Then mutation breeding, it is creation of the variability, then making the selection. Gene changes you are making, right? Sudden change in the heritable characters, you mean the genes are changed and they are inducing that changes and then going for selection. So these, mutation breeding. So if you just look at all these methods, as I explained, there are methods suited for self-pollinated crops, there are methods suited for cross-pollinated crops, and there are methods for population improvement, and there are methods for handling the segregating method. This you should, if you remember this, then you will not get confused with recalling what is the procedure involved in each of the plant breeding method. This is very important. So we can just discuss these plant breeding methods in future classes. For crop improvement, there are two important steps or requirements rather. There are two important requirements. One is variation must be there in the base population, right? It should be now variation. Some changes are different genotypes are there. Then only you can do something. You can identify the best. Selection does not create variability. It only utilizes. Remember this. If you go for selection, means you are not going to create any new things. It is just you are observing the best one, identifying the best one and pushing it. That means there should be difference in the plants or the population. Some different plants are there, then only you can separate them out. Oh, something is good, something is bad, like that. If everything is same, then no, the selection is not going to be effective, right? So that is must. That means variation should be there in the population. And then this variation, whatever you are observing, it should be heritable. It should not be non-heritable. Heritable only, there is a meaning for the selection. But the good one, what you are saying, and then pushing it for the next generation, or it will, perform better. But if it is because of the environment, it is because of, not because of the genes present in that plant, then it will not, be expressed in the next generation wherever you are evaluating it. So that means whatever that change in the variation is there in the population, that should be heritable. These are the two important requirements for, the plant breeding, right? For the improvement. How to know that the observed variability is heritable, whether it is heritable or not? For example, some good plant you see is a very high yielding one, and then the cornet. If we collect the seeds and put it in the next generation, if it is not doing well, what does it mean? Next generation. That means it is because of that environment factor, or that soil or fertility or watering is given good to that plant, and it is performing well. But it is not because of its inherent capacity. So it is required progeny test, right? You collect the seeds from particular plant and raise it in the next generation. If the progenies are doing well, then only what the selection you have made in the previous season is meaningful. Otherwise, it's a waste, right? So for that, progeny testing is required. So, so what is progeny test? We have already given you the definition of that. Evaluation of the worthiness of a plant on the basis of performance of their progenies is known as progeny test. Not its own performance. Progeny test is, right, the progenies, its offsprings, how they are performing. Based on that, the parent's ability is decided. So that is progeny test, right? Evaluation of the worthiness of a plant on the basis of performance of their progenies is known as progeny test. So, you may have already told you that Louis D. Vilmorin, it is also referred as the Vilmorin isolation, who has postulated this Vilmorin isolation principle or Vilmorin principle. He employed it for improvement of sugar content in the sugar beet, right? So that is the progeny test, how important. So it is, variability should be there, and existing variability should be heritable. To know that whether it is heritable or not, progeny test is required. Okay, that's what, the basic requirements. There are two basic steps in crop improvement that also you should remember. The two basic steps of crop improvement are creation of variability. First, you have to create variability. If it is already available, you need not. It's simply use. And second step is selection. These are the only two steps, most important basic steps in any of the plant breeding method. Creation of variability, second is selection. Some methods here, they are not going to create variability, it's only selection, right? Yeah. So, but by and large, all the plant breeding methods together, there are only two steps involved for the crop improvement. That is, first is the creation of variability, second is selection. So it doesn't mean that both these steps are involved in, but selection is there in all the methods, whereas creation of variability is there or not, it depends on the plant breeding method. So selection is important. Selection means, what do you mean by the selection? The definition of the selection: permitting only plants with the desirable characters for reproduction. As far as plant breeding is concerned, how you have to define? Permitting only the plants with the desirable characters for reproduction. That means you are identifying the good plants, and only seeds of those plants you are collecting and raising it for the next generation, raising it for the next generation. That means you are permitting only the plants with the desirable characters for reproduction. Okay, so that is the basic one, that is selection. And the selection also, like you can classify the selection process as natural and artificial. Natural means, what do you mean with natural? Naturally, you just grow the plants and allow. In modern plant breeding, this has a little value. That means, like, the bulk method of breeding, natural selection can apply. Suddenly, it just simply raise the population. Some good will contribute more seeds, and poor will contribute less seeds. That means, that is also taking care, the selection. Or else, if you grow some population in the high temperature, the plants which are resistant to the temperature, tolerant to that high temperature, they will survive, others will die down, right? Natural, or some disease is there. The plants which are going to be, the resistant to that particular disease will survive, others will vanish. That means there is natural selection. But you are leaving it to the nature only. Whatever it happens, what disease it comes, what temperature it comes, water, humidity, all that you are leaving it to the nature. But it has a little role in modern plant breeding, because you want to do it faster. Otherwise, you cannot wait for the nature to enforce the selection pressure. The artificial selection is very important. Current plant breeding entirely depends on this. So it essentially depends on the phenotype of the plant, and its effectiveness depends on the reflectivity of the genotype, the phenotype. So what is that artificial? You are going for the selection. That means you are identifying by your naked eyes, right? You are just looking at the plant and you are making the selection. So it essentially depends on the phenotype. That means phenotypically, right? What outside it appears is very important. Based on that only you are picking that, you are choosing the plant, right? And that effectiveness of that selection, right? It depends on the reflectivity of genotype in the phenotype. Meaning, phenotype means externally what it appears. Genotype means what the genes it carries. So phenotypes should reflect what it has got inside. It is not because of the environmental factors like more water or fertile soil or the good agronomic practice if the plant is doing well. So that is not going to help. It is only if the phenotype if the phenotype, if you look at it, it is a reflection of the genotype, then only it is going to be effective. So that is, the selection basic you should understand. Then artificial selection, there are three types of selection you can just, based on its effects or directions. Mathers 1953 as classified the selection. It's important, basics you should understand. You can use those things for crop improvement. So there is the classification methods, classification directional selection or unidirectional selection. Selection favors at a time one of the two extreme types of individuals in a population. Mean of optimum genotypes is different from the population mean. So it's a revolution force. What is that directional selection or unidirectional selection? The selection, what you are pressure you are putting it favors the at a time one of the two extreme types. For example, if you have got a plants, from uh, 0 to 100 scale, any character you take it favors either 100 or near 100 scale plants or 0 near 0 plants. So the unidirectional, right? One direction. And the mean of the mean of optimum genotype is different from the population. If you take the 100 near, you take the mean of those selected plants, and if you take the mean of the entire population, definitely it is different, right? Even 0 to 0 near 0, whatever scale the plants are there, the mean of that is lower compared to the overall population mean. So that's what the base population is there, mean of that, and the selected population mean, it is different. Mean of optimum genotypes is different from the population mean. It is a revolutionary force. It's changing to one direction, right? Then there is a second type of selection, that is stabilizing selection. Here, mean of population does not change, but variance changes. Right? Mean will not, right, change. It remains same, whereas variance changes. So weak stabilizing selection tends to increase variance, while a strong selection reduces it. So the mean remaining unaltered, it is a conservative force. What is that you are going to do here? Plants, whatever you are selecting, the mean of that particular character, if you take it, that will be same as original population, also new. What is that? That's why it is stabilizing selection. Whatever it is, observation is seen, it is over and and again, it remains same. That is important. Whereas variance changes. That means that both the sides, extreme types of plants, you are removing. Only the center, the mean, near to mean plants, you are retaining. Right? It is a stabilizing one. So the weak stabilizing selection tends to increase the variance. Right? We are not too much concentrated on stabilization, then there can be increase in the variation because of the recombinants. While strong selection, very high, very few plants you are going to select. So that means selection, while the strong selection, it reduces the variance. So, but mean remains unaltered. It is a conservative force. So this, type of how you are. So there is one more selection type, right? You have got directional selection or unidirectional selection, then stabilizing selection, you understood. Then you have got a third method, it is a disruptive selection as defined by Smith 1962. Disruptive selection favors those members of the populations which are above or below of the average population, right? So what is that? That means near to 0 also you are selecting the plants, and near to 100 also you are selecting. Those populations and those individuals are selected, and further, they are pushed to the next generation. So disruptive selection, it favors the members of the population which are above or below the average populations. Now, if you just look at this, the graph of a population for a given trait, it is drawn, this curve we have got. Stabilizing selection, only this is favored. Whereas directional selection, only this is favored or this is favored. Whereas disruptive selection, this is also favored. This is also favored. This is also favored. These two are favored. Whereas these are eliminated. So this is what ultimately your entire selection process involves, what you are going to do, right? It is the directional selection or unidirectional selection. It is, the stabilizing selection, and it is the third one is the disruptive selection. These are the very basic selection methods. Mather has given the classification. So, okay, that's what you should remember for any plant breeding method you go for. You can use those skills of selection, right? What do you want to do it? Whether you want to go for stabilization, you want to go for extreme types, or you want to retain both extremes. So there can be some intention for doing that. So pure then, so then we'll let us start discussing on the individual plant breeding methods. One of the plant breeding methods very important for the self-pollinated crops, that is pure line selection. So it is pure line theory. A pure line is a progeny of a single homozygous plant of a self-pollinated species. Therefore, all the plants in a pure line have the same genotype. What do you mean by the pure line? A pure line is a progeny of a single homozygous plant of self-pollinated species. So one plant which is self-pollinated, from that seeds are collected and raised. Those plants referred, as a pure line. So pure line is a progeny, right, of a single homozygous plant. Because it's homozygous, it will not segregate. Same two type you'll get, but because of its self-pollination. So this is applicable to the self-pollinated crop only. So pure line is a progeny of a single homozygous plant of a self-pollinated species. Therefore, all the plants in a pure line have the same genotype because there is no segregation, right? Because the plant, what you have selected, mother plant, is a homozygous. From that, any number of plant, you take it, and it is the self-pollinated crop. Remember the rule. That's why you have got that homozygosity there, complete homozygosity, and you are carrying it further. And the plants in a pure line have the same genotype. There is no change from one plant to other plant. All have same genotype, one genotype. So phenotypic difference is anything, externally if you look at those plants, progenies, phenotypic differences within a pure line are due to the environment and have no genetic basis. The genetic difference is not there. It's only because the environment, phenotypically, the plants are differing from one another. Therefore, variation within a pure line is not heritable. It is because of the environment, no heritability for that variation. So within a pure line, no, you cannot, apply selection. Right? A pure line means it's all the plants are alike. All the plants are homozygous. All the plants have got the same genotype. Right? Any difference you are seeing morphologically, it is because of the environment, and that is not heritable. Right. So it dates back to mid 19th or even earlier. That is from 1840 to 1860. Several workers, notably D. Coulter, Sheriff, and Hallett, and Wilmore, they practiced in individual plant selection in crops like wheat, barley, and sugar beets. They did it, individual plant selection. What we said, it's a separate plant breeding method. Individual plant selection is not a separate plant breeding method. It's, you can generally say it's pure line selection. They thought, right, if it's a cross-pollinated crop also, they did it, but it has to be self-pollinated. Then only it is going to be effective, because all the assumptions you have drawn, it is a homozygous, you said it. Why homozygosity is there? It's because of the self-pollinated crop. Several generations it is self-pollinated, it's homozygous, and there is no change in it. So this is used enormously in the 19th century. The concept of pure line was proposed by Johnson in 1903 based on his studies with French bean and used to improve the seed size of the French bean. Origin of variation in the pure line selection, you said it, and where the origin comes here, the variation comes here. So origin of variation in pure line, with the time, right, that variation comes. How with time, how it comes? It is mechanical mixture. Some mixtures mechanically come. Or natural hybrid is a very rare. If you say the self-pollinated crop like tomato, very highly self-pollinated, or any other crop you take, it is less, up to 5% cross-pollination is there. Even it is a self-pollinated crop, tomato. So that means that natural hybridization also can occur, right? And it can give. Otherwise, it's not easy to get a variation within a pure line. As the time passes, natural hybridization can be there, and the mechanical mixture during handling, that can also bring the variation in the pure line. Self-pollination increases the homozygosity with the corresponding decrease in the heterozygosity. Similarly, inbreeding increases the homozygosity and reduces the heterozygosity. That you know. So inbreeding you use for cross-pollinated crops, self-pollination is used for the self-pollinated crop. Yeah, there are other ways by which the variation is within the pure line you will get. That is the chromosomal aberration. This is also like the aberrations can happen at the time of reproduction of the cells. It can happen, sexual part involved cells, and then mutations. Yes, mutation also can happen. If it takes time, it's not very frequent. It's a rare. So these are the four ways how you can get the variation in the pure line, right? Pure line, mechanical mixture, variation within the pure line. Passes of time only you'll get, not immediately. But how you get? Because of mechanical mixture or natural hybridization or chromosomal operations or mutations. It's a kind of, the in vegetable crops, pure line selection method is adopted to improve the heterogeneous foundation. Heterogeneous foundation stocks. That means variation is there. Each plant is homozygous, but they are different from one another in a foundation stock. And as a result, varietal improvement is possible. That's why you carefully use the terms. Population, when you use it, should be in the cross-pollinated crops. But if you want to use the same way in the self-pollinated crop, you should not use the term population. You should use the term foundation stock or heterogeneous material, right? It is not the population, because individual plant is not heterogeneous in a self-pollinated crop. Okay, that's very, very important. Yeah. Characteristics of a pure line. Now, some more things we can just throw light on how this pure line is. All the plants within a pure line have the same genotype as the plant from which the pure line was derived. And the variation within the pure line is environmental and not heritable. And pure line becomes genetically variable with time because of all those reasons, like the mechanical mixture or natural hybridization or mutations or the chromosomal operations and all that. So these are the characteristics of a pure line. Whereas what are the uses of pure line? What can you make use of this product of this pure line selection? It can go as a variety, and it can be used as a parents in the hybridization program. In some studies like mutation, because there is no genetic variation, mutation study is very important. In studies and mutations, avoid genetic variation in the experimental material. Any change, genetic change is because of the mutation only. So if you use already variated material which is having the variability, and you go for mutation studies, you don't know whether you have created the variability because of the mutation or it is already there. You don't know. But whereas if you use the pure lines, you will know that any genetic variation is there, it is because of the mutation. So in studies on mutation, this pure lines can be used. Other studies avoids the genetic variation in the experimental material, and the true effect of treatment can be detected. So other studies, wherever you want to have some experiments, the genetic material itself is variable, then the effect of treatment you will not be knowing. So in that case, if you use this pure line, there is no genetic variation. You are getting it's only because of those treatments, the variation you can get. So experimental material, whatever you are using, and true effect of the treatment can be detected if you use the pure line. So these are the uses of pure line. Now the application, the improvement of local varieties like Pusa Savani and Bendi, then improvement of introduced variety can follow this pure line selection. Improvement of old pure line, because time is passed, variation has accumulated there because of the mutation or chromosomal aberration or mechanical mixture or natural hybridization that you want to use. And then selection for a new characters in a pure line. Earlier, that character was not considered as improvement. So if one pure line, you can use it for, one particular character, which was not important at the time of release of that variety, but now it is important. So there also the existing pure line, it can be used. Selection in segregating generations from crosses. The pure line selection is same procedure. Selection, if you make in segregating generations of process, that is purely for self-pollinated crops. Otherwise, cross-pollinated crop, it keeps on crossing. It's only on selfing, and the homozygosity it gets. And the meaning of that pure line selection, yes. What are the general, the procedure for pure line selection? Yeah, general procedure. lot many modifications in procedure and number of years is possible. It's not the fixed one. Here season or the year and what you are going to do in the each of the season. First season, large number of plants selected from local or mixed population. Carefully use the term population. We should not use foundation, right? Based on the characters identified. And the variation is must in base population. Certain plants are homozygous. They are self-pollinated because it's a self-pollinated crop. And identified characters are few, not too many characters you can use. Visually identifiable and heritable. What the character you want to improve, there should be, you can visually see, and those characters should be heritable. So 200 to 3,000 or less number of plants can be selected from this, depending on the resources and the crop, how many can handle. And now the second season, progenies of individual plants are grown separately with proper spacing. So one row means here, if you are selecting 200 plants, means first plant, it should go as a separate line. It's really not the progeny test because its presumption is that they are pure lines, no mixture. But if some segregating one, it can be identified. So progenies of individual plants are grown separately with proper spacing, drastically reduce the number of plant progenies to be carried out for the next generation, because it is simply you cannot have too many. You have to save the land, labor, time, money. Everything is required for this. So you have to apply the selection here also. Segregating progenies you have to discard unless they are promising. So by chance any homozygous plant is there and they have got the plant, that will segregate, right? So those plants you are rejecting unless it's very promising. Then disease epiphytality can be created, right? And naturally, those plants can be, whichever are showing the symptoms, you can, but if every plant is, the susceptible, then you have to select some good based on the yield or other characters. Superior progenies are harvested separately, right? So now individual rows are there, means individual plants, you are harvesting them separately to carry it for the next season. So third season, now immediately, the very third season, you have got preliminary yield trial for a critical evaluation. Now PYT, the preliminary yield trial. So earlier, just visual only you had done it. Now yield records you have to make it. You have to record the yield. Use the best variety as a check. So here, along with the variety only, existing variety, you should evaluate it. If anything is better than the existing variety only, it's meaningful. Otherwise, use best variety as check. Further reduce the number of progenies. If you have got more than 100, I have to reduce it. Of course, all those which are above the check variety. Then 4th to 8th season, there is replicated yield trial, RYT. It's very common. It is here without any replication. One line means only one replication was there. Whereas from here onwards, it will go as a 3 replication or 4, whatever number of replications you have decided. So that is replicated yield trial. Specifically, you are targeting for the yield. So then coordinated yield trial, you are giving it to the different stations and you are getting the information. Or multi-location trials, coordinated yield trials, multi-location trials, you have to do it from 4th to 8th. So that means regularly, repeatedly, the selected few varieties are assessed here, right? So observations on disease reaction, you have to take flowering, maturity, and other characters. No. At the time of release, you need all these characters. Now you have reduced the number. The varieties should be very few to go at finally 4 or 5, right? Multi-location trial also, it should not be more than a dozen varieties. I mean, the lines. So within that, you have to have the description of the varieties which you are pushing for the release, further process. So 9th season, best strain or a progeny is released as a new variety. And you need to multiply the seeds. You have got a very few plants there. You have to supply the seeds of that particular line, large area. So you have to multiply the seeds. So that is what the general pictures. So it's the same thing, just in the brief one slide, just. So activities 200 to 3,000 plants are selected on the basis of their phenotypes first year. And then individual plants, whatever you have selected, the seeds are collected separately and raised in the separate rows. So individual plant progenies are grown. Undesirable progenies are rejected here also, maybe repeated if necessary. And of course, anything is segregating or removing unless it is worthy. And then third year, it is the preliminary yield trial, right? Remaining progenies are planted in preliminary yield trial. Inferior progenies are rejected here. And then the fourth to sixth year, multi-location trial, like that RY, replicated yield trial. Replication, not one block, it is same varieties, repeated replicated yield trial, conducted at several locations. Inferior progenies are rejected, disease resistant and quality test can be done to describe the variety. It's not that you are straight away going for resistant variety. You get right, most of the time you won't get, but you have to say that these are the diseases which this genotype is susceptible. These are the diseases to which this varieties, this pure line is quite, the tolerable. Like, so best progeny is released as a new variety. So seed multiplication distribution. So you have to multiply. Seventh year itself you can do it, or season. You can if you want to go faster, you can have more seasons per year, and you can reduce the time. So what are the advantages of pure line? Achieve maximum possible improvement over the population used, right? The best one plants you are selecting. Variety is extremely uniform and liked by the growers. Uniformity is there. And marketing media, men, consumers, everybody likes the uniform, right? Same mixture is there, people will not like, right? So it is then easy identification of variety. If the population is there in cross-pollinated crop OP variety, some variation is there. It is difficult to identify whether this variety is same, what I am proposing or not. Here, I mean, variety ultimately it is released. So easy identification of the variety possible. Then disadvantages of pure line, poor adaptation. It's not only one genotype is there. If more genotypes are there, at least few can adopt like that, the product of your mass selection and other things. More expensive it is because need more time, labor, money, land compared to some other methods. Then upper limit is set by the original population, foundation stock, whatever it's there, based on that only, it's upper limit. Breeder need to devote more time because individual plants are selected by the breeder, visual observations. So examples of pure line selection are tomato variety Arka Vikas and Arka Saurabh are the results of pure line selection. Like that, we have got, the different institute, the variety, how it is developed also. Arka Vikas selection from the tip top indeterminate root medium. The description of the variety is there. Arka Saurabh selection from the variable breeding line V685. Then Arka Abha AVRDC collection VC8121 subjected for selection, resistant to bacterial wilt. Then Arka Alag, it is also resistant to bacterial wilt. It's a pure line selection result. So Pant T3, Pantnagar, JB Panta Agriculture University, Pant University of Agricultural Sciences and Technology, semi-determined, most suitable for winter season. S120 IRI selection from the Nahu lines, dark green foliage. Then in brinjal you have got CO1, MDU1, CO2. Then purple long, Arka seal, Arka Kusmakar, Arka Srish. They're all the results of pure line selection. Okay. So what are, sometimes, these descriptions are given wrongly in some of the references. You have to be careful of that. So what is that originally the plant breeder says, what method is that we have. Then G5, 4, G5, G3. In Chile, I doubt, of course, they said it's a pure line selection, we have to take it, but it's a cross-pollinated crop. This is what in the literature I had to get the information from. So the problem with these vegetables means it is not really handled by the professional plant breeders. That's why this variation in use of the terms, and there can be some confusion, but they don't know the plant breeding accurately, precisely. For that reason, they described this method. Really, they have not followed that method. Many times it is questioned, right? K1, CO1, NP46A, Musalwadi, Arkalohit, all are the products of pure line selection. That's what the plant breeders, at the time of release, they mentioned what method you have followed. Same thing there. Okra, Pusa Makmali, CO1, and Gujarat Bhendi. And in winged wing, IHR selection 2160, not promising simply. Similarly, WBC2 from Meghalaya, good. Of course, then Narka Komal, French bean, Narka Bold, Cornell 49-242. Then Dolichos bean, Arca J. Like, these mentioned as the varieties developed through Q-line selection. I'm not getting into the, the controversy, some corner, some varieties are mentioned, derivative of pure line selection, and some references mentioned as the pedigree method. That all depends on from where, which source. It is originally what the plant breeder says that we have to take it. Next is the, single plant selection. I said it's rather than a separate plant breeding method, it is just a chance factor. A single plant selection is used to select a chance variant of types, superior types from a mixed homozygous population in self-pollinated vegetable crops. This is one of the effective methods to make maximum use of germplasm. You are not having a separate program for that. Somewhere, germplasm you are multiplying or you are handling, maintaining. At that time, you have something good, you got it. Oh, suddenly you feel that it's going to go as a variety. You can. So that's why it's a single plant selection. Then you subject it for the further procedures like preliminary yield trial with check, and then replicated yield trial, multiplication trial, and then you can multiply the seeds and release it. It's a variety. Okay, that is a single plant selection. Like tomato, you have got Liana, Greater Baltimore, and Bonnie Best. Broad bean, you have got Encore from USDA collection from Afghanistan, best adapted to Canada. Then you have got a clonal selection. I was talking about this, the selection process. The clone is a group of plants. Clone is group of plants produced from a single plant through asexual reproduction. It is, the clone, several plants you are producing from a single plant and asexual method. What is that? That means they are two trait. Same like pure line selection also here, whatever mother plant is there, same thing is their approaches. But the difference here, there in pure line, the mother plant is homozygous, but it need not be here in clonal selection. Whatever the mother plant, because the two type you can produce, but whether it is homozygous or heterozygous, you don't know. For clonal selection, wide range of, to make the selection, again, variability is required. So for that, what you have to do, several clones you have to collect. For a clonal selection, wide range of clones from various sources are collected. From this collection, superior clones are selected. Unless, the variation is there, you cannot make any selection. Second year, these are grown separately and superior clones are selected on visual observations of morphological traits. Third year, these are tested along with local checks. Fourth year, replicated yield trial. You need not have to spend more time for this clone because they are, the clones, they are vegetatively propagated, or just comparison is there. Best clone selected is multiplied and released. Example, potato, Kufri Red is clonal selection from the variety Darjeeling Red Round. Red Round, why one variety? How you get the variability? So Darjeeling Red Round, it is there from several, the decades. During the passage of time, so many decades or even century, you can get some variability and your selection is effective. Similarly, the Red Round and Kufri Safed are selection from the variety Fulwa. Sweet potato Vikram from ABRDC collection. So these are like, the examples of pure line selection. So with that, we are, ending up on the first, methods of breeding, general description, and then pure line selection, single plant selection, and clone selection.